Hello, everybody, and welcome to this Effective Altruism for Christians guest speaker event. Our speaker today is Ana Karina Pitol on the topic of pandemic preparedness. Welcome, Ana, and the floor is yours.
Thank you. Just 1 second, I'll share this. Can you see? Can everyone see and hear well?
Yes.
Okay, well then I'll start. Well, my name is Ana, and today, the presentation of today is very general, very basic presentation, a brief overview of pandemic preparedness. And yes, just 1 second. Okay, here. So just first of all, a little bit about myself. I work on understanding the transmission of infectious diseases. I did my PhD focused on enteric viruses, which are mainly viruses that are responsible for gastrointestinal diseases in low-income countries. And then when I was doing my PhD, I actually came across effective altruism. And through some meetings of effective altruism, I got interested in schistosomiasis, which is a disease that affects around 200 million people in the world. And I moved to schistosomiasis research, and I work and continue working in understanding how can we move from treating people sick with the disease to eliminating the disease in some countries. And then COVID-19 hit, and I moved a little bit to work with SARS-CoV-2 and with other pathogens of pandemic potential. And what I do is, so at the beginning of the pandemic, what I worked on was understanding the risk of interacting with contaminated surfaces. At the beginning, it wasn't clear whether it was risky or no. Now we know that it's not the main source of transmission, so people shouldn't be so stressed about it. But yeah, when you have questions such as what's the risk of touching a contaminated surface, there are different data that you need in order to arrive to an answer. It's not just how long does the virus survive in different surfaces, but also how long it survives in skin, how much it transfers from one surface to another, and so on and so forth. And that's kind of like the kind of research that I've been working on lately. And today, I'm not going to talk about my research, but I wanted to talk a little bit more about pandemics, because that's a topic that it's more interesting to people belonging to the effective altruism community. And I gave a presentation a couple of years ago before COVID-19 about pandemics and biosecurity. I remember like 20% of the presentation was introduction, which is completely unnecessary now. I think we all know what a pandemic is and why it's important. Just kind of like a big reminder that ever since humans have been around, there have been infectious diseases causing epidemics and pandemics. And that is reflected in literature, like from like myths of before Christ to, stories like Romeo and Juliet and so on. They have like, like part of the background story has a little bit story of the consequences of having a pandemic. And there have been some devastating ones, like, for example, the Black Death, the bubonic plague, killing around half of the population in some cities, and the 1918 influenza and so on. But I think by now we're a bit aware that these pandemics just happen to happen every so often. And back in the days before the COVID-19 pandemic started, there were already some pathogens that were highlighted by researchers working on emerging infectious diseases as pathogens of pandemic potential. For example, the WHO had a list of 7 pathogens that were called the priority list or the blueprint list. And here I'm putting 4 of the ones that they have in the list. And as you can see, one of them was the coronaviruses, MERS and SARS coronavirus. And the reason being that coronaviruses and other viruses, they tend to jump from different hosts, from one host to another, and that increases the risk of they actually getting to humans and creating an outbreak that could lead to an epidemic and further to a pandemic. And we have always been concerned. There was not a lot of people working on pandemics, but there was still a number of people working on emerging infectious diseases. And there were some worry things that will worry people working in infectious diseases. Some of them, it's how connected we all are right now because of globalization and the higher population sizes that make the spread of diseases just easier. The closeness to animals. We have, for example, factory farming, where we have just confined in a very small space, lots and lots of animals that sometimes have outbreaks. And these outbreaks can lead to a jump between, a jump from the pathogen from the animal to human. And other factors like climate change. But we also had some stuff in our favor, which is that as compared with, for example, the bubonic plague, if the bubonic plague, if the pathogen responsible for that will arise now in a community, will be better equipped, and it wouldn't lead to 40 to 50% death rate because we have better diagnostics and we can react faster to these concerns. So anyway, just to say that before COVID-19, we already had a bit of an understanding, and we had some things in our favor and some things playing against us. And for the people of effective altruism, the cause area of pandemics and biosecurity became quite interesting, but not so much because of natural arising pandemics. That's an area that, as I mentioned, it wasn't completely neglected. There were people working on it. We knew a little bit the risk, and so it wasn't really because of that people from effective altruism became kind of like interested in that. But it happened mostly because of something called global catastrophic biological risk that could be a huge pandemic caused by humans. And there are two ways humans can cause pandemics that could potentially be more deadly and worse than natural-made pandemics. One is by accidental release of pathogens, and then the other one is bioterrorism. And in this presentation, I would like to explore both of them a little bit. So first, let's start with the accidental release of pathogens. So the question is, should we be concerned about having an accidental release of pathogens, and how concerned should we be? And my answer will be, yes, we should be concerned, mainly because of two things. There are many researchers doing research that is considered dangerous, and I will talk a little bit about this. And then accidents happen all the time in these laboratories, and I'm also going to talk a little bit about this. So first, let's go for talking a little bit about what kind of research we will classify as research of concern. A bit the story of doing research with biological agents. In the Cold War, it became, let's say, trendy for some countries to have laboratories where they will create agents to use as bioweapons. But back in the 70s, the Biological Weapons Convention was formed, so it was created. And the goal of this was to prohibit the development, production, acquisitions, transfer of biological weapons. And it was quite successful. So some countries that had laboratories where they were, producing smallpox or anthrax and so on, they closed those laboratories, and we advanced a lot, moving towards having a more safe world without all these pathogens. But after this, we're still doing a lot of research that could potentially be dangerous. Some of this research is called gain of function research, which is basically you modified, for example, a virus so that this virus becomes either more transmissible or more virulent, or you can also make it move from one species to another, so kind of like enhance the capacity of that pathogen of concern. And there are quite a number of laboratories that had carried out this kind of research and continue to carry out this kind of research. One example of this research was the one with influenza virus. In 2011, there was a Dutch scientist called Dr. Fouché. I don't know how to pronounce it, Fouché. I'll call him Fouché. And well, what he did was, in his words, he mutated the hell out of H5N1 virus to make it achieve mammalian transmission. So maybe I'll explain you first a little bit about this virus. This virus, it's an influenza strain that is transmitted between birds, but it's normally not transmitted to humans. But sometimes people who work in close contact with birds will get this H5N1. And actually, I think we had the first case of H5N1 influenza in human in the UK a couple of weeks ago. But anyway, these are weird events. They don't happen so often. And the good news is that once the person is infected, the person is not capable of transmitting the virus to another person. So it's just an isolated event, and this person doesn't create an outbreak. But the bad thing about this virus is that the death rate of this particular strain of influenza, it's around 50%. So it will be really bad if this influenza actually becomes, mutates so as to be able to be transmitted from humans to humans. And that's basically what he wanted to do. He wanted to see whether that was even possible. So what he did, in his words, was mutating the hell out of the virus. So it kind of shows how lack of, I don't know, I would say like concern he had of this kind of research. But anyway, so the thing is that he managed to do it, but I mean, of course, not using humans, but using mammals. He used ferrets, and he managed to mutate the virus so as that a ferret can actually transmit it to a different ferret. And they have been, after that, there were a couple of articles also working with gain of functions research, some of them actually funded by funding bodies in the US. And there was a lot of concern, and these concerns lead to regulations happening. So in 2012, like just shortly after all of these experiments got public, the regulations were more strict. So they required that people doing these kind of experiments will have to go through a different route of approval, of biosafety approval. And what they were asking them to do, it's a balance between the benefits of the research and the potential risk of the research. And basically they had to justify that the benefits were way bigger than the risk of the research. But the reality is that after this research, there have been a lot of more research on this gain of function kind of experiments that have been published without going through this route of approval. And one of them, for example, is in 2014, there was another scientist who modified this pandemic strain of influenza, again, to try to either discover whether it could be more transmissible or more virulent and so on. And later on, it was understood that this person did apply, so did ask to the biosafety committee for permission, and the biosafety committee didn't classify the research as dual use of concern, which is dual use of concern means when something can be used either for good or for bad. And it seems that even the people responsible for evaluating the biosafety procedures weren't capable of identifying the risk in order for them to actually classify it as a dual use research of concern. And a couple of years later, there were some kind of like attempts at understanding how many of these kind of research are actually following the guidelines and the route, and it was less than 10%. So in one paper that was analyzing this, the author says that it is likely that the research community is just unable to recognize research as dangerous due to lack of training or proper guidelines. And this became, again, a topic of conversation now with coronavirus, because we still don't have anything conclusive about where the virus is coming from. So the WHO went to China, as you probably know, and they did an investigation, and then they say, oh, it was more like the most likely scenario is that it just jumped from an animal to a human. And it is probably that's what happened. Spillovers from nature happen all the time, and you do have a lot of coronaviruses in animals around the area of Wuhan. SARS coronavirus 2 closest relative, it's found in the bat, and the genome, it's 96% identical to the one of SARS-CoV-2 that we have in humans. Now, the problem with this is that 96% identical, it's not close enough as to you be able to say that it just jumped from the bats to the human. So the theory now is that it jumped from the bats to another animal, and then from another animal to the human. But until you find the other animal and you find a version of the coronavirus that is closer to SARS coronavirus 2, then you cannot really prove that this theory is correct. And there have been a lot of attempts to actually sample animals from around the area to try to identify this animal that could potentially be the host of this SARS coronavirus 2. And yeah, the thing is that they cannot completely rule out the hypothesis that it could have been leaked from the laboratory, because the Wuhan Institute of Virology was carrying out research on coronavirus. In 2015, there was an article, I think in Nature, where they basically say that research carried out in that institute on gain of function research on coronavirus was risky. And then they kind of explain why that research was particularly risky, and the institute perhaps wasn't as well equipped to deal with that research. And also the fact that there is a distrust on China, because at the beginning of the pandemic, they just they hide some information and so on. So there's also this level of like, we should trust them that they're saying that it wasn't them, but it's kind of like hard sometimes for people to just accept that they're just saying the truth. So anyway, we have these two hypotheses that are still open, and this, I think, highlights, regardless of what the answer is, it kind of highlights the possibility of having laboratory leak of a pathogen. And talking about the possibility, I would like to also bring to the attention of everyone that accidents happen more often than perhaps we're aware of. Like, for example, if you just Google biosecurity incidents, Wikipedia will give you like 51 major incidents, and that's just the tip of the iceberg. You have incidents, for example, scientists getting infected. This is just random selection. Getting infected with SARS from BSL-4 laboratories in Taiwan, in Beijing, sometimes even spreading it to other people outside the laboratory. And you have, for example, the US Army lab mistakenly shipping anthrax, live anthrax, 74 times, dozens of labs. You have laboratories from United Kingdom leaking the pathogen that caused the foot and mouth disease and causing an outbreak because of broken pipe work. And I could go on and on and on. An accident in China that caused 65 workers to become infected with brucellosis, and later on, these workers actually infect people in their town, and they have now an outbreak of that. And anyway, I suppose with this slide, I just wanted you to be aware that it's not an isolated case. I've worked in labs of biosafety 1, 2, and 3, and I've seen accidents in the 3 of the biosafety labs. And most of the time, those accidents aren't really even reported. So yeah, so it is a bit concerning. Now, we move away from accidents. I hope that I kind of made clear the general idea that it's a possibility that these things happen. And we enter into bioterrorism. Now, bioterrorism, it's something that I think was more of a concern before, and then it kind of stopped being so much of a concern, and then it started being a concern again. But it's just hard to estimate the risk. In a paper back in 2015, they compiled a lot of expert opinion on the topic. And there was this meeting where they put together a lot of the leading people in biosecurity in US. And they invited them to participate in a study. And they asked different questions. In here, I'm just putting one of them, which is, what do you estimate to be the likelihood of a large-scale biological weapon attack occurring within the next 10 years? And as you can see, you have answers from 0 to 100, literally. So there is really no consensus. There is no pattern. It's like a uniform distribution of answers. And that is because it's really hard to estimate these things. People are not trained to come out with estimates for these questions. But if we were to try to answer this question of estimated likelihood of a bioterrorism attack, you will have to know a few things, but 2 things mainly. One is, how many bad people do you have around? And bad, I mean, yeah, like how many people that will be interested in carrying out a bioterrorism attack, and how many people have the skills to actually go on. Now, if we just assume that the number of people interested in such an activity will remain constant through time, we can still get into a little bit diving into who has the skills for doing something like this. And this is a quite an interesting question. And the answer, I think, is more and more people every day. Because first of all, we have a lot of information out there already. We have something called information hazard, which is that when scientists publish things that could be used for harm, like, for example, sequences of pathogens. And we have a lot of sequences already available of many different pathogens. And you also have scientists trying to publish things like, what makes viruses jump from one species to another, what makes a pathogen more viral and more transmissible, and so on. So we're just kind of like making everything public, because we have this, tendency of like wanting to share so that we can move forward and advance in our understanding and therefore our fight against diseases. But this could also backfire because there is a lot of information out there. And then what do you do with that information? Well, the reality is that we have the technology to do these viruses from scratch. For example, in my lab, when Omicron came to the scene, we wanted to have Omicron in the lab to study. And we considered having it through 2 routes. One is through patient, so you have to wait and apply and get the samples from a patient, or you can actually make it yourself from scratch. So this, as of today, still requires a little bit of technical skills, but it's just becoming more and more feasible for more people to do this kind of stuff. And in order for you to do it from scratch, you will need the pieces, the sequence of these virus, for example, in this case, the Omicron. But you can be trying to do smallpox or something like more deadly. And what you need is pieces of these, in the case of Omicron, for example, pieces of this genetic material that you will require to actually create the virus. And those pieces, you normally get them from companies that synthesize those segments. And these companies, the good news is that they actually scan those sequences. And if you have something, a code that matches the code for, let's say, smallpox, then they wouldn't be synthesizing that sequence for you. So they have a system to scan from them. And these arise from a consortium these companies, the biggest companies of gene synthesis, they got together in 2019 and they create this consortium to safeguard the biosecurity. And I mean, I think this is a great thing that they do. And the good thing is that the companies that are part of this consortium are basically the responsibles of synthesizing around 80% of all the segments that get synthesized in the world. So it's good that we have this level of protection, but I think it's also important to have in mind that, for example, this effort was led by the companies themselves, not by a regulatory agency from not for the WHO, not by any government, because when they started to make this, there was not regulation for that. And so I think it's a good effort, but technology, it's just advancing very fast. And now we have ways to even synthesize these materials in-house. So for example, this is just one example of a desktop size automatic DNA synthesizer. And the whole goal of the company who makes this is to empower research so that, every biologist have the in-house capacity to do their own pieces of DNA or RNA. And you also have new kits being created every day to make things easy for you. For example, this kit says that it's ideal for customers new to gene synthesis. So I guess this is just kind of like a bit of example of how fast we're moving, and and how much these kits help the person who is doing this kind of research to achieve the goal of creating whatever they want to create. I think back in the days, these kind of complex processes to create, for example, a complicated plasmid that will be going into a cell to create a virus, will be something that only a very, very, very skilled person will do. But we're moving towards pretty much anyone in high school being able to do these kind of things. So if we go back to estimated likelihood of a bioterror attack, we can just start to notice that it could increase with time, because the skills necessary are just going to decrease, and the money necessary and everything necessary to actually do it. So I'm coming to an end, and I do not like to end in a negative note. I think there has also been a lot of positive things, some of them actually coming from coronavirus pandemic. Some of the positive things is that we are capable of surveying people and the environment and animals and so on in a way that we've never done before. For example, wastewater treatment plants are now up to date with sequencing technologies and sending their samples to sequence to detect coronavirus for sure, but they're increasingly detecting other pathogens, and they know now how to do that. And that's new. I mean, that's new for most wastewater treatment plants. Also, there is an increased concern of these pathogens that are jumping from animals to humans. So there's increased emphasis in surveying these animals as well. And we've invested so much money in diagnostics, and it's paying off. There are technologies that are just really good that could, in a couple of years, mean that a person could give a sample, and then this sample is run not only for the pathogen of concern, but for thousands of pathogens, and you will have the answer of the presence or absence of thousands of pathogens. And that's because of this huge push that money brought to the field of diagnostics. And then we have sequencing becoming every time more cheap, and with this cheapness comes the possibility of sequence more and more samples. And for sure, we had a huge success with therapeutics and vaccines. The fact that we could create the RNA vaccines so fast gives us hope that in the future, if another pathogen arises, we could react even faster. And there is a really nice episode in the 80,000 Hours podcast with Andy Weber as a guest, who is the Department of Defense member. He was, I think, in the past. And he was kind of like making the case for investing a lot, not so much on prevention, but on all these countermeasurements that could be used both in natural pandemics, but also in bioterrorism or accidental release of pathogens. Because the more we are prepared, the faster we react, the more we're able to contain this, then the less appealing it will be for someone to actually get into creating a pathogen and spreading it. If it's just less appealing because we have the weapons against this other weapon, basically. I don't know if, and yeah, so I think with this, I would like to conclude, but I'll be happy to continue the discussion.
Great. Thank you so much, Ana. I will keep the recording running just a few minutes longer because we have a couple of questions there in the chat. So maybe if you can answer those first, and then we can turn off the recording after that, and we will have room for people to ask with their own voice, so to speak. So yeah, the first one is from Ray. And by the way, if I'm reading your question in the chat, if you want to clarify it, or if you have a short further question, you feel free to jump in, though I remind you that the recording is still running, but if you don't mind that, that's fine. So the first question in the chat is from Ray. Could we differentiate leak of sample from gain of function research?
Yeah, so perhaps I can clarify the question. If a sample is being collected in a cave and the agenda is to protect workers, and then the person collecting the sample becomes infected in an accident or in the laboratory, that's very different, I think, from a leak, from an experimental gain of function, which is then deliberately or accidentally released. And I didn't hear you making that difference, and that's quite important for the Wuhan case. Also, the branch of the virus that they were working on was nowhere near SARS-CoV-2, you know, and it was a long time back. It's really hard to make that connection. It would be much easier to make a connection with South China or North Vietnam, for example, and there have been samples there.
Yeah, I mean, I think I agree with what you just mentioned, but so was it there still the question of whether we could differentiate?
A different question. I felt you didn't really make that clear. You had it all together as one. And,
yeah,
and
yeah.
The difference between someone accidentally getting sick, doing a brave thing to protect other people in a cave, versus someone irresponsibly doing an experiment.
I think it's not someone irresponsible doing the experiment. It's someone doing the experiment and then having an accident happening, which I think it just can happen to everyone. Even, I'm not saying like, even the people doing gain of function research have like a malice intended or anything. They want to do something good. It's just that they accidentally either get sick themselves because, I don't know, sometimes they're working with animals and then the animals bite them or something like that, which is also the case. I agree that there's other kind of research where the researchers take samples from bats in caves and then it can happen that they get infected, and then by that releasing the virus. Whereas with the question of if there is a difference, so there are some modifications that people do in some of these gain of function research where you can see some kind of like traces, because there are some enzymes that you can use that kind of like give a little bit of like a trace, but some techniques don't leave a trace, and some traces are also available in nature, so it's not conclusive. So I think it could be used to discard, but I have heard arguments saying that, for example, in the case of SARS-CoV-2, that there are some traces of something that could have been an enzyme, but then other scientists say, oh no, but you also can find that in nature. So I think it's still, I think if it's too obvious, something too not natural, then you can say, okay, it was a gene that it was inserted there that shouldn't have been there in the first place, something like that. But also, gain of function research, it's kind of like a very general term, but it can also mean you having, like, for example, in the case of influenza, you having the influenza strain and putting it into a mammal, in this case, ferrets, and putting a lot of ferrets together, kind of like trying to passage through ferret until it changes. So in this case, it changed in a way naturally, but it changed because of this kind of research. So I don't know if
that was the first one. I think there's one from Katarina.
Mm-hmm. Yes. So the next question in the chat is are you more concerned about states or individuals / groups making bioterror agents?
I think I'm personally more concerned about individual groups, individual people or groups. I'm always concerned. So when I go to the lab, I'm like, I don't know why they trust me. I don't know why they trust my colleagues, like even going inside the lab and for me thinking that I could just take it out, it's not like there is cameras or anything. So I'm concerned even about the scientists working on that. It's like, what if someone decides that they don't want to live anymore, and then I don't know, humans are causing a lot of pain, and they could just easily take anything from the lab and then just put it in the distribution system, water or whatever in Tesco on. So because I'm thinking of that all the time, I'm normally more concerned about people because I feel like we are just too many, and sometimes someone gets kind of like crazy. And I think like the cheaper this becomes, the easier this becomes, the more available this becomes, then the higher this concern will be. Whereas with countries, it kind of stays relatively this, like if there is no like a, I don't know, if huge wars or something like that. And even if there is, they wouldn't do biological, they wouldn't use biological weapons. They have other means of more targeted damage.
Thank you. And next question in the chat, who is doing the data work to predict return spillovers of SARS-CoV-2?
So predict return spillovers?
So we've got a lot of copies of SARS-CoV-2 in the human population going all over the world. And in this year coming now, we're going to have a huge number of copies of the virus, including in zoonotic hotspots, South China, Brazil, Mississippi, blah, blah, blah. Mm-hmm. Lots of opportunities for the SARS-CoV-2 to go into animals. We don't even know how many species. And then it can come back like it did from the mink in Denmark, but that was contained, which is great. But isn't there rather an elevated risk of a pandemic this year, a whole new pandemic because of a recombinant variant coming back from an animal species?
That's a good question. And I don't I don't have like a certain answer to that one because I haven't
Let's
thought
get together
about it
afterwards
so
and see if we can find them.
I think for me it makes sense, the fact that you have, more hosts that could potentially, the virus could potentially mutate. But at the same time, I don't think that's normally what happens I'm just thinking in previous pandemics and
in humans, so I'm guessing it will happen in animals as well. It just needs an immunocompromised deer or tiger or mink. So just that there are so many copies. I think I'm just assuming the background risk of a pandemic this year must be higher because of that. What I'm not sure is it like, have we gone from 3% to 4% or from 3% to 20%? That's the.
So you're talking about something that is significantly different than SARS-CoV-2.
Yeah, so something on the way to MERS or SARS-1.
Because what happened normally is that, there's a lot of mutations happening, but then there are some strands that are just way better than others, and we see it all the time, right? And it really takes over. So it can also happen that it just becomes endemic, and then you have like the strong strains, and then we have more of a situation like the case of influenza that.
Yeah, I mean, I'm personally more worried about a Delta, a recombinant Delta variant, recombinant Omicron. But, it would be nice to exclude this possibility.
Yeah, I don't think you can ever exclude these things.
I mean, if you can work out that it's genetically not possible, then you can exclude it.
Okay.
Then maybe we should look out for it and try and stop it really, really early this time.
Yeah, I think there are a lot of efforts to stop things early. I think that's kind of one of the take-home message from this pandemic. So there's a lot of survey happening, and there's a lot of sequencing happening. But this sequencing is happening in developed countries. So
that's my concern, that we might miss North Vietnam, Colombia or something.
Could be.
Okay. The last question in the chat is, are there any public charities with room for funding that you think may be highly effective at making progress on this issue?
I really don't know, because this issue, because there is so many things that go hand in hand with the current pandemic. I think there has been a lot of funding. Like funding, hasn't been an issue. It's not really neglected. And that's why we observe so much progress in the areas of diagnostic and treatment and so on. So I'll have to think more, and if I come back with it, maybe I can just directly text Michael, because from the top of my head, I think like it's kind of like one of these exceptions where money it's not the limiting factor here. I think now we have money.
I think the exception is the zoologists who are exactly working on the spillovers, crossovers. So it would be worth checking in with WWF if they're working on that. And one of the people who went into China from the WHO was interviewed on BBC Science podcast recently. So I'm pretty sure that you would be surprised how little funding some UN organizations have got. So I would imagine they could, yes, in fact, I know that department was cut before the West African Ebola outbreak. That was one of the problems. So yeah, the WHO would probably take your money if you've got some.
What are your thoughts on the failures of the model of an internal review board versus making that more of an external responsibility? I think one of the challenges, having sat on a few of them, is there is a pressure to approve your colleagues' work. And it's subtle. I think most scientists, if they saw something clearly dangerous, would be moral enough to put the brakes on it or ask for more guardrails. But I do think those edge cases where you're doing something that sort of maybe it's dual use, maybe it's not, happens kind of a lot. Do you have any insight on sort of reforming the internal review board?
I don't have, I mean, I have the same concern that you have, because, I mean, for those who aren't really familiar for how this works, normally the people reviewing this, it's people working within the same institute, normally scientists, basically. So as, I mean, as Matthew mentioned, it's just your colleagues, and you have already the bias that you want this to be published, you want to move, like you don't see the risk yourself. So I think like that's a really good point. And I didn't have, because this is not really my area of expertise, biosecurity, so it was kind of like a lot of new stuff for me as well. But I did wonder the same, whether there have been some efforts put to kind of like have it outside, that it's not the institute. The only thing I can say is that in the countries that I have worked, the problem is the same. So all of them have the internal board reviews, and I'm not aware of any big, but that doesn't mean that there is no big efforts happening right now that I'm not aware of. It's just that I don't know.
Okay.
Thank you.
Yeah, thank you.